Hello everyone, my name is Rachapun and I'm currently a graduate student at the Okinawa Institute of Science and Technology. And in this video, I will be presenting my work entitled Experimental Exploration of a Ribozyme Neutral Network using Evolutionary Algorithm and Deep Learning. So firstly, a finite landscape is a conceptualization of the sequence space of a biomolecules, such as RNA, DNA, or protein, as a function of its fitness. And proxy for fitness can come in many forms, including catalytic activity or antibiotic resistance. A neutral network in the context of finite landscape is created by sequences that are connected through single-step mutation that share the same phenotype. And neutral networks have been extensively studied using theoretical models, especially in the RNA sequence structure map. These maps use predicted RNA secondary structure as proxy for fitness. The studies that utilize these maps have revealed that neutral network can facilitate evolution, leading to higher mutational robustness and can influence evolutionary adaptation and innovation. Yet, not a single work has experimentally demonstrated the existence of such network. In this work, we try to find such evidence through the use of high-throughput screening and deep learning guided evolutionary algorithm. We chose to study the sequence space of a 35 nucleotide catalytic core of the F1 style U ligase ribozymes. This RNA enzyme can catalyze the ligation of two RNA fragments in a template-directed manner. Because this is an essential process for cell replication, this ribozyme has important relevance for the origin of life and the RNA war hypothesis. Our method involved the design of successive generations of ribosome variants using in silico genetic processes, which include selection, mutation, and recombination. So briefly, each generation starts with a commercially synthesized pool of ribosome sequences, which were then experimentally screened to measure the activity related to the Y type. These sequence activity datasets then undergo in silico selection, mutation, and recombination to design the new population, and the process are repeated for 6 generations. The final generation, or generation 8, were designed with a combination of deep learning model, which was trained using data from the preceding generations. The model was combined with the in silico genetic processes to form a completely in silico evolutionary algorithm. This plot shows the fraction of neutral mutant which we designate as any variant with at least 20% of the Y-type activity. The fraction of neutral mutant increased drastically in generation 6 when the in silico genetic processes have been optimized. Then we train a number of models to classify sequences into neutral or not using data from generation 1 to 6. As you can see, all models perform relatively well, with the MLP model, or the multi-layer perceptron, having the highest recall. So then we chose to experimentally validate the performance of the MLP in combination with the in silico genetic processes. Generation 7A was made using only the in silico selection, mutation, and recombination, while Generation 7B also had the MLP model to select only sequences that are predicted to be neutral. You can see that the use of MLP increases the fraction of neutral mutant to almost 90%. Finally, we used a complete evolutionary algorithm to create Generation 8, which were able to identify neutral variants with around 27% accuracy. However, looking at the distribution of Hamming distances of the sequence in each generation, we can see that the average Hamming distance in Generation 8 is significantly higher than other generations, meaning that the evolutionary algorithm was able to identify neutral variants at high Hamming distance from the Y type, despite being trained only on lower distance sequences. Furthermore, we identify a mutant in Generation 8 that has 16 mutations compared to the Y type, while still maintaining comparable activity. We suspect that this mutant might be connected with the Y type through an extensive neutral network. To confirm this, we experimentally screened the entire combinatorial library, so all 2 to 16 variants that connect the mutant and the Y type through single-step mutation. We found that indeed this combinatorial space is well populated with neutral variants, and many neutral paths exist that connect the 2 variants. Furthermore, we found that neutral variants within this space can be accurately predicted by the MLP model that were used to design Generation 8 without any additional training. So why is it then that the neutral network that we discovered were able to be predicted by the model more easily than other regions of the finite landscape? To investigate this, we performed the Walsh-Hammadath transform on the dataset, which essentially convert the finite values into the background average epistatic terms. And the terms include all orders from 0 to 16. After doing this, we found that the fitness value in neutral network can be easily predicted using only information from lower order epistasis, where significant predictive power can be achieved using only third or fourth order epistasis. So in conclusion, we found that deep learning guided evolutionary algorithm can be used to explore neutral regions of the RNA finite landscape by leveraging the high predictability of neutral network within the landscape. Thank you.